There's so much to discover about multiple sclerosis. MS attacks the brain, spinal cord, and optic nerves of patients, causing pain, fatigue, vision, and coordination difficulties. The cause is unknown, and treatments are investigated. Through it all, great strides and success in the research of MS are recognized and awarded.
We want to know what causes MS. I don't have any illusions that I'll ever figure that out, but I hope that my colleagues around the world as a whole will get this figured out within a reasonable period of time.
The professor of neurology at Washington University School of Medicine in St. Louis is among a prestigious group of researchers. Anne Cross was awarded the 2019 John Dystel Prize for Multiple Sclerosis Research. She's one of 2 women to receive this high honor out of 25 people in the world who share it.
That list of 24 people preceding me is quite a list of important people who've made a lot of important contributions. I did have some very good female mentors. I think as the generations go on, there are more and more women taking up this role.
Cross won the award for her pioneering work into immune cells known as Bcells, then later advancing new imaging techniques detecting MS activity and exploring possible benefits of calorie restriction to reduce signs and symptoms of MS. Her journey was accompanied by many influential colleagues, beginning with the pursuit of Bcells. Cross was first to demonstrate the critical role of Bcells in an animal model of MS.
I worked on trying to get a B-cell-centric model, animal model for MS. We found that if we tried to make the disease in B-cell deficient mice that had been made to not have B cells genetically, couldn't get the disease.
This led Cross to the next step, studying B cells in people with MS, which was a direct challenge to what was already widely pursued, that MS was caused by immune cells called T cells.
There were many more T lymphocytes in MS lesions than there were B lymphocytes. So again, that was sort of sweeping B cells to the side, and people lost interest in them.
But Cross did not.
For a while I had been thinking, if we could get rid of B cells in humans, we could figure out how important they might be in the real disease of MS. So when I heard about the drug rituximab that depletes B cells from humans, FDA approved for non-Hodgkin's lymphoma, which would be a B cell cancer, then I thought, well, maybe we can use it off-label in MS.
This led to a human study in 2002. Cross showed how B cell depletion can help those suffering with MS by reducing relapses and lesions in the brain.
Well, the results were kind of unbelievable. They were 88% reduction in gadolinium-enhancing lesions in the 3 after treatment MRIs. So they didn't develop new lesions or new attacks down the road on these MRIs.
Although she had success, that drug was never FDA approved for MS for reasons involving potential side effects. But her study helped pave the way for the development of a new B-cell depletion drug, now approved as a therapy for MS.
Ocrelizumab since 2017.
So her work isn't done, which is why she's now part of a multi-center study that includes 100 participants.
It will help us understand MS if we know better, how it is actually working in people to reduce relapses. And in my study, we did spinal taps before and after treatment. So in this bigger study, which is multi-center, we're doing the same thing and looking at the changes in spinal fluid as well as in blood, but do it much more in depth and use our, the cutting edge things that we have now.
The link between B-cell depletion and clinical improvements remains the focus of her research.
We don't know how getting rid of B cells in the blood, as well as in the spinal fluid, which is what these agents do, how that really is creating this benefit. We don't know how many years we can safely continue to B-cell deplete people.
In the study, B cells may have center stage, but something is happening with T cells.
About a 50% reduction, 50-60% reduction in T lymphocytes, which these drugs don't go after T lymphocytes. So that is another mystery.
Cross is also helping to pioneer a new imaging technique called diffusion imaging to track and measure nerve damage in MS patients.
For many years, I've been working with Xinghuai Song, who's a PhD chemist, radiologist in our radiology department here. He and I, many years ago, somehow got together and started working on diffusion imaging. It's very, very high resolution, microscopic level, can look into the central nervous system non-invasively, won't hurt people, safe. And so that's how we got together.
Cross says the imaging is currently at the research level and says it's as good as a biopsy, which is something that's not commonly done for MS.
There are many different lesions in people's brains, so even if you biopsy this one, it might not be the same as this one. So what I was looking for was a non-invasive biopsy, shall we say, of the central nervous system where we could follow people over time and see how it changed and understand what was going on as close to the microscopic level as possible,
which can help determine best treatment options for MS patients. For HEC, I'm Kathleen Berger.